My research group in general is interested in one of what I consider to be the remaining challenges in childhood acute lymphoblastic leukemia, which is really how best to manage and treat leukemia that's spread to the central nervous system. So that's the brain. And we know that although leukemia originates in the bone marrow, if you don't actually give treatment directed towards leukemic cells that might have spread from the bone marrow to other sites in the body, and particularly to the brain, that somewhere between 50 and 75% of children will have what's called a relapse, a recurrence of the leukemia within the brain itself. And for that reason, all children currently receive quite a lot of chemotherapy that's actually specifically directed to the brain. And although that's quite effective, the rates of relapse in the brain are relatively, thankfully, low. We know that therapy is associated with really quite a lot of toxicity, and we at the moment don't really understand very much about CNS leukemia, the types of treatments that we might need for it, and how to limit that toxicity. So our research group works basically on all those aspects of therapy against CNS leukemia. The grant's really exciting, I think. What we're trying to do is identify children who are at risk of what we call toxicity from the therapy, so neurotoxicity. We know that probably somewhere between 20 to 50% of children can be susceptible to having some adverse effects of this treatment on things like their ability to concentrate at school, verbal reasoning, processing speed, etc. But at the moment, we really can't pick those children out at all. So it doesn't seem to be particularly related to the dose of chemotherapy you get or any other factors that we know about. So some children sail through therapy. We have plenty of children who are cured of leukemia in childhood, are now doctors and lawyers and university professors and do fantastically. But then there's a smaller group of children that really seem to have some adverse effects of their chemotherapy. So this grant is really trying to see if we can identify ways of picking out these children who are having these adverse effects really early on, at a time where we might be able to prevent them or produce new treatments to try and make those things better. And also whether we then might be able to pick out children who require different types of treatment that might not have so many adverse effects on their learning and memory. The first thing we want to do is really say, can we pick out these children that they're going to have the problem, or that have the problem really early before it actually causes them a problem? The second thing we have to do is to do something about that. Now, this particular grant is not going to give us an instant fix to that, but it's really trying to tie in not just can we pick these children out, but can we see why they're having the problem. So we're actually taking samples of what's called the cerebrospinal fluid, which is the fluid around the brain, that is actually routinely sampled when these children get their treatment. So the CNS-directed therapy is actually giving lumbar punctures, which is spinal taps, and injecting the chemotherapy directly into that fluid. When we have that needle in, we always take a sample of the fluid as well, just to look at under the microscope. And we're taking the leftover sample of this, and then we're looking within that to see if we can find out what might be causing this problem with the toxicity in the brain. So we're looking for markers of damage to cells in the brain, but also specific what we call metabolites, which are the breakdown products related to the chemotherapy we've given. And we have really good evidence from what we call preclinical models, so not in patients, but in experimental models in the lab, that there might be particular metabolites, these proteins that we find in this fluid, that actually will act on a receptor that might cause this toxicity. And the really exciting thing about that is we actually have antidotes to these molecules. And one of the antidotes that seems to work in these preclinical models, so it's not been tested in children yet, is actually a common component of cough mixture. So it's a medicine that's routinely given to children, that appears to be safe, and that will, in theory, kind of neutralize the adverse effect of the chemotherapy without actually affecting how good it is at fighting the leukemia. So the breakdown product that causes the toxicity is quite separate from the way that the medicine actually kills the leukemic cells. So we, obviously are super excited about the idea that might actually be something that in the future we might be able to give these children a dose of cough mixture before we give them their chemo, and that might allow the chemo to work but not have the toxicity. But that's a long way off. We have to test that in proper trials. So I'm not saying this is something that's just around the corner, but this study and this funding enables us to really test whether what we've found in the lab is actually true in patients as well, which is a really vital step before thinking about new therapies.